Okay. All right. Good morning, everybody. Let's get started. So today we're going to finish the digestion absorption of nutrients, and then we talk part of carbohydrate metabolism. So we've already finished protein, carbohydrate digestion absorption last lectures. So and today we're going to talk about the lipid digestion absorption. So first question is dietary lipids in the great amount in our food. What type of lipids do you guys think is the most abundant in the food? Which type of, yes. Triglyceride, yes. Thank you. Triglyceride is the most abundant lipids in the food. So we deal with a lot of, triglyceride after we eat. And in addition to triglyceride, there are also other type of lipids in the food, like for example, cholesterol esters, phospholipids. And before we talk about digestion of the lipids, particularly triglyceride, I want to show you the triglyceride structure. You guys probably already know, right? Just wanted to refresh your memory. So triglyceride is composed of two parts, right? One is the glycerol backbone and has 3 carbons, and then the other part is fatty acids, free fatty acids. Three fatty acids attached to 3 backbones, 3 carbons of glycerol backbones, right? So glycerol backbone here, and you have 3 fatty acids attached to the glycerol backbone and form the triglyceride molecules. And 3 carbons of glycerol backbone, we named the 3 different positions. So top one is called SN1 position, middle one is SN2, and the bottom one is the SN3 position. And you can see probably already, different position has the different type of fatty acids attached to it. And mostly the fatty acids attached to the SN1 position mostly is the saturated fatty acids, right? And SN2 position fatty acids is unsaturated fatty acids. You only have the 1 double bonds. And the third position most likely is the polyunsaturated fatty acids. You have multiple double bonds, right? So this structure of the triglyceride is very important for our better understand how the enzyme digests triglyceride molecules. Again, the glycerol backbones. And just some more structures for other type of lipids like cholesterol and cholesterol esters and phospholipids. So phospholipids basically is similar to the triglyceride, but the difference is that at SN3 position, the fatty acid is not the fatty acid, but the phosphorylcholine group, for example, in this case, attached to the SN3 carbons of the glycerol, right? And cholesterol esters, if you compare to free cholesterol, differences is again is the cholesterol esters has 1 fatty acids attached to the free cholesterol molecule, right? So R group represents fatty acids molecule, right? So attached to the cholesterol to form cholesterol esters. And also I wanted to tell you the concept, the difference between the free versus esterified lipids, right? So free lipids, for example, free fatty acids, and fatty acids and cholesterol. These lipids are called the free lipids, right? And once the fatty acids attach to glycerol backbone, forming triglyceride, and triglyceride is the esterified lipids. And same thing for the cholesterol esters, right? Cholesterol ester, as I mentioned, one free amino acids attached to the free cholesterol molecules and forming cholesterol esters. So cholesterol esters is also considered the esterified lipid, right? Triglyceride, cholesterol esters are esterified lipids. Is that clear? Right? All right, so lipid digestion. First questions, where does lipid digestion occur? In which part our body starts to digest lipids? So digestion triglyceride begins in the stomach. About 10 to 25% of lipids or triglyceride are digested in the stomach by an enzyme called gastric lipase. So gastric lipase is secreted by stomach and digesting triglyceride to break down the triglyceride molecules into this fatty acids and glycerol backbones. And the rest of the triglyceride digested in the small intestine and by a different enzyme. The enzyme also the lipase, but secreted from pancreas, right? So stomach and pancreas are able to secrete lipase and digest triglyceride. So in two different parts, right? Different places, stomach and small intestine, and by different enzymes secreted from different places. And gastric lipase usually preferentially cleaves the SN3 position of triglyceride. Remember that 3 carbon positions on the glycerol backbone, right? So means that gastric lipase specifically cleaves the free fatty acids at the SN3 position of a glycerol backbone and release that fatty acids, right, from the triglyceride molecules, and generating 1,2,1,2 refers the position of the remaining 2 fatty acids molecules at the glycerol backbone, right? So 1,2 diacylglycerol and 1 free fatty acid. So this free fatty acid is released from SN3 position, right? Once the triglyceride digested by gastric lipase. All right, does that make sense? Okay, so now pancreatic lipase and cleave the fatty acids from the different positions, SN1 and SN3 positions, right? So release fatty acids from SN1 and SN3 position of a glycerol backbone, and generating 2 monoacylglycerol and 2 free fatty acids. Right? So pancreatic lipase, it seems more powerful. I can cleave or can break down the 2, cleave the 2 fatty acids and release 2 fatty acids from the triglycerol molecules, and generating one. It still has the 1 fatty acids attached to the SN2 position, right? So, and how phospholipids and cholesterol it's digested? Again, it's also in the small intestine, but by different enzymes. So the phospholipase A, which is secreted from our pancreas, and phospholipase B is secreted from the brush bottle membranes, and these two enzymes is responsible to hydrolyze the phospholipids into fatty acids and the lysophosphatidylcholine molecules, right? So just try to remember the phospholipase A and B are the two enzymes that digest phospholipids, right? Release the fatty acids and the other phospholipid molecules. Cholesterol esterase is an enzyme which also secreted from pancreas and digests or hydrolyzes cholesterol esters to free cholesterol and the fatty acids. Remember, cholesterol ester, this molecule is composed of two parts, right, molecules. And free cholesterol and the one fatty acid attached to free cholesterol. But the cholesterol esterase is also be able to digest the triglyceride and ester, vitamin A, D, and the monoglycerols. But the main function of a cholesterol esterase is to digest cholesterol esters, right? Okay, the lipid absorption. So lipid absorption, if you guys still remember, last class we mentioned that there is a water layer on the top of a brush border membrane, right, in enterocytes. And because of the existence of this water layer, lipids cannot be, themselves cannot be efficiently absorbed into the enterocytes because lipids are the water insoluble, right? So if nothing else, only lipid molecules themselves, you can see very inefficient, right? Very few molecules of lipids be able to absorb it into the cells. So our body actually secreted bile acid in the small intestine, and this bile acid or bile salt combined with the lipid digestion product to form micelles or vesicle structures, see here. And this will allow the hydrophobic portion of the lipids buried inside of the micelles and only allow the hydrophilic portion of the molecules exposed, right, outside. And hydrophilic portion is better be compared to the hydrophobic part, is more water soluble, right? So this micelle or vesicle structures and be able to allow will allow the lipids more easier to be absorbed, be easier to pass through this water layer and be absorbed into the cells, right? So this will facilitate more lipids be absorbed or more efficient absorbed into the cells. So bile acid is very important the molecules in our body that can facilitate lipid absorption. So lipids transport, after digestion, are transported into the enterocytes. I just mentioned, right, through the micelles, or sometimes we also call liposomes. Liposomes is similar. It's similar concept, right? It's the micelles. Sometimes we also call it liposome. So the concentration gradient across the brush border membranes that facilitate simple diffusion, right? So the mechanism, how lipids is absorbed into the cells just through the simple concentration-dependent fusion mechanisms, right? So concentration gradient is very important. That means that you have a high levels of lipids in the intestinal lumen and lower levels of lipids inside of cells, so across the Rush-Bottle membrane. So that concentration difference will push more lipids being absorbed into the cells, right? So simple diffusion is the mechanism for lipid absorption. And another thing is very important. Once lipids being absorbed into the enterocytes, and the free lipids will immediately re-acidify to acidified lipids. So this mechanism will allow the free lipids, keep free lipids at a low concentration inside of cells, right? So that can maintain high the concentration gradient. So allow the more lipids being absorbed or transported into the enterocytes. So once the free lipids being absorbed into the cells, there is the set of a protein called fatty acid binding proteins. So immediately binds free lipids and then transport free lipids to the ER. And this mechanism is also again to facilitate to maintain the low concentration of free lipids inside of cells. All right. Any questions? And cholesterol has their own binding proteins, right? It's called sterol carrier proteins. That, yeah, question. Oh, yes, sorry. Yes, endoplasmic reticulums, right? It's organelles inside of cells, right? Usually, for the triglyceride synthesis, it happens in the ER. So, so the fatty acid binding proteins transported free cholesterol from the place where they absorbed it to the ER for the synthesis of triglyceride. Triglyceride again is the acidified lipids, right? So, so two monoacylglycerol and fatty acids can be reconstituted or reacidified to form triglyceride, as I just mentioned, in the ER, right? And the phospholipids can also be reacetylated to form phosphatidylcholine, or hydrolyzed to form 3 phosphorylcholine, right? And transported to the portal vein. So as similar to the free fatty acids or triglyceride, free cholesterol also can be reacidified to cholesterol esters, right, by this enzyme we mentioned before, a cholesterol esterase, and also another enzyme, but you don't have to remember this enzyme, right? You just try to understand this kind of process, how the cell need to do something to keep the concentration gradient, right? And to allow the more lipids being absorbed inside of cells. And something is different from the carbohydrate and protein transporter or absorption is that the lipids, once it's absorbed into the enterocytes, not directly transported to the portal vein, to the circulation, but to the system we call the lymphatic system first, and then to the circulation, right? So lipids absorbed and transported to the lymphatic system first, and then the circulation. This part is different from the absorption of carbohydrate proteins, right? Another important thing is that the lipids, the exported from the small intestine to the circulation, or to the lymphatic system, and finally circulation, is through the is mediated by the set of protein called lipoprotein. Have you guys heard about lipoproteins? Right. So lipoprotein has several different types of lipoproteins. Right. We're going to have a specific session about this later. So lipoproteins are the set of proteins that can carry lipids and transported the lipids from absorbed small intestine to the other tissues, organs, through the circulation, right? And most important lipoprotein that synthesized in the enterocytes in small intestines is chylomicrons, right? And also some small amount of VLDL. VLDL is very low density lipoproteins, right? So again, the chylomicron is the most abundant lipoprotein that's synthesized in the small intestine that can carry the triglyceride and other cholesterol and other phospholipids from small intestine to the other tissues. And liver adipose tissue are the final destination of the absorbed lipids. Right. Any questions? And clear? Okay. So this is the diagram. I think it probably will help us to better understand this whole process. You know, the lipid digestion, absorption. So dietary triglyceride again is the most abundant lipids in the food. So when we eat food and we may get a lot of triglycerides, so we need first of all digesting the triglyceride. And there are two enzymes, right? Most important enzyme, the gastric lipase and the pancreatic lipase, right? And just by name, you know that these two different enzymes secreted by two different places, right? Organs like stomach release the gastric lipase, and pancreas release pancreatic lipase. And these two enzymes that cleave or digest triglyceride is differently. It differently means that, you know, the enzyme cleave the fatty acids from triglyceride molecules at the different carbon positions, right? And once digesting, the final products of the digestion by these two enzymes are the free fatty acids, 2 molecules of free fatty acids, and 1 molecule of the 2 monoglyceride, right? Does that make sense? Okay, so so once it's after digestion, the free fatty acids and the 2 MAG can be absorbed, of course, through the mechanism they called. Do you guys know the very important mechanism for lipid absorption, the structure? Micelles, right? And how micelle is formed? Bile acid, right? Bile salt and lipids itself forming the micelles. And micelles is very important structure that facilitates lipid absorption into the enterocytes, right? So once it's, and again the mechanism to facilitate the lipid absorption is what kind of mechanism that facilitates lipid absorption? Simple diffusion, right? And simple diffusion is based on the concentration gradient, right? The lipid concentration across the brush border membrane. So we know that after meal, we also after digestion we also have a high concentration of free lipids in this side, right? In the lumen, and a lower concentration inside of cells. And in order to keep this concentration gradient to allow more lipids absorbed, and the mechanism is very important. So once the free fatty acids or 2-MAG, 2-MAG, I would just call it MAG, right? So 2-MAG is absorbed into the cells, and this free fatty acids will immediately resynthesize to triglyceride, and in this way, then this free lipids concentration will go low, right? Does that make sense? So and again, I wanted to mention to you that the fatty acids always need some protein to help move from one side to the other side. So we have a called fatty acid binding proteins, right, to help to bind the free fatty acids and help the move of free fatty acid to the ER for the synthesis of triglyceride, right? So once triglyceride synthesized and triglyceride being packed into the lipoprotein, and in the small intestine, chylomicron is the most abundant lipoproteins, right? And so the lipoprotein and then released into the lymphatic system, right, and eventually to the circulation and to the other tissues. So so this is, I think this diagram is pretty good to help understand the whole process, right? Absorption and transporter to the body system. Yes. Actually, triglycerides are synthesized on the ER. So once you absorb it from here position, right, it's not the ER yet. So but you need the fatty acid binding protein to help the free lipids to move to the ER for the synthesis of triglyceride, right? Okay. Any questions? Yes.
What percentage of the free fatty acids enter the portal?
Oh, yeah, the good question. So free fatty acids, not like a long chain fatty acid, but like medium or short chain fatty acids are able to direct being absorbed to the portal vein, right? Not through this mechanism. So only short chain or medium chain fatty acids can be directly absorbed into the portal vein. All right. Okay, so this slide I just wanted to remind you that just make a comparison, the digestion absorption, the difference between the carbohydrate, lipids, and so in terms of the location, carbohydrate and lipids both can be digested in the lumen of the small intestine, right? And protein digestion, in addition to the small intestine, protein can also be digested inside of an enterocyte. Remember this, right? Because enterocyte can secrete or produce the important enzyme called amino peptidase and carboxylpeptidase. And these two enzymes basically is responsible for digestion of di and tripeptides once they absorb into the enterocytes and to the free amino acids. And lipid digestion is complete in the small intestine, and the transported into the enterocyte through the diffusion mechanism, but need the micelle structures, which compose of the bile acid, bile salt, and the lipid products. And the important process, the intracellular transport by fatty acid binding protein and the re-acidification back to the triglyceride. This mechanism actually have two purposes. One is to keep a free lipids concentration low inside of cells, right, to allow to have a higher concentration gradient across the brush border membrane to facilitate more absorption. And the second purpose of the re-acidification or triglyceride synthesis is to allow the triglyceride being packed into the lipoprotein and transported out of the enterocytes to the lymphatic system and eventually circulation, right? And in terms of the transporters, remember the carbohydrate, glucose and amino acids absorption requires transporters, right? Protein transporters, right? Glucose transporters and amino acid transporters. But lipids is not. Lipids is just a simple diffusion, but of course it requires the micelle, structure to help the diffusion. And lipids, yeah. Okay, so those are kind of, the differences between the carbohydrate and lipid digestion absorption. All right, any questions about lipid absorption, digestion part? Okay, so now we finish the All right, so let's start to talk about the carbohydrate metabolism. So in this session, we just wanted to learn the general concept of a glucose metabolism, metabolic pathways, glucose, how glucose is metabolized by different pathways, and how our body be able to maintain the glucose homeostasis. Homeostasis means that we because our glucose level always be maintained at the very narrow range, right? And we during the feeding or fasting or starvation, our blood glucose can be flux a lot. So, but our body has a system or has a mechanism to not allow the glucose levels to be too high or too low. So, and how our body can make it. So, we also wanted to learn this kind of mechanism, so-called glucose homeostasis. And also important thing is that for glucose metabolism, the glucose transporter into the different tissue is also very important and also controlled by the hormones. For example, insulin, right, is the most important hormones, can regulate the glucose transporter into different tissues in the different physiological states. And finally, glucose metabolism in the specific tissues, that means glucose can be metabolized differently in different tissues. For example, in the liver can store the glucose in the form of glycogen, right? So glucose being absorbed into the liver can be synthesized to glycogen. And liver is the only tissue, liver and muscle, right, are the most important tissue that can synthesize glycogen and store glycogen, and not the other tissues, right? And for the okay, so here just try to give you an example. In a different tissue, they metabolize glucose differently. So we also need to learn, different these differences. And also for the glucose metabolism, we wanted to learn in the different physiological state, in the fat state when we eat food, right, or during the fasting, or when we don't eat food, and our body metabolize glucose also differently. For example, in fat condition, we have a lot of glucose, right? We get a lot of glucose from food, and we cannot use all of them, right? So we need to store some glucose as stored energy and use it for later during the starvation. So the glycogen synthesis will be very important. Pathways are being activated during the fat condition when we eat. And during the fasting, and we wanted to use them stored energies, right? So break down glycogen. So glycogen breakdown pathway would be very active during the fasting. And in addition to the glycogen breakdown, the gluconeogenesis, our body can also make a glucose, right? So the gluconeogenesis pathway can also be very active. So again, different tissues metabolize glucose differently, and different physiological conditions, glucose can also be metabolized differently. So with all these general concepts in mind, then let's start to talk about it. So digestion, absorption, and transport of carbohydrate we already learned, right? And what about the final product of digestion that from the carbohydrate that our body can absorb into the circulation and deal with it? What are the final product of digestion? Remember, of carbohydrate. Glucose, fructose, right, and galactose. And again, because of the amount, the glucose is most abundant. So we mostly talk about the glucose metabolism, right? And glucose is the most important cellular fuels. And in addition to glucose, there are other called cellular fuels, energy fuels, are, you guys already know, right? The glucose and fatty acids, ketone bodies, amino acids, they are all the energy fuels, mean that these molecules can be oxidized in the cells and produce ATPs, right? And ketone bodies, usually we don't get ketone bodies from the food, right? But our body makes ketone bodies. And not only at any times we make ketone bodies, only during the fasting that our body make ketone bodies. And the reason why we make ketone bodies, we'll get there in the later sections, right? And also our body can make glucose from the non-glucose precursors. For example, glycerol, lactate, and propionate are the important precursors of glucose. So during fasting, our body can also make glucose from these non-sugar precursors, right? And glucose, again, is the primary metabolic fuel in humans. Provide 32 to 75% of energy in the diet, based on what we eat. So glucose is a primary metabolic fuel, right? And all tissue cells are able to use glucose as energy fuels. So any type of cells are able to use glucose as energy fuel. I say that because some other energy fuels, for example, fatty acids, cannot be used by some specific type of cells. For example, red blood cells and brain cells. These type cells are not able to utilize fatty acids for energy fuels. Why? Just keep this question in mind, all right? Okay, so and physiological state I already described this, that when we learn glucose metabolism, we need to thinking about in the fat and fasted condition, right? Fasting condition. So glucose is how glucose is metabolized depend on the physiological conditions, right? And no matter in fat or fasting conditions, the main goal of our body to metabolize glucose is to maintain the circulating glucose levels, right? All right. Do we want to take a break? Okay. All right. So yeah, we can come back about 11:32 or 33. Right. Okay. All right. Let's move on. Um, so, so our body metabolizes carbohydrates. We try to understand how our body metabolize triglycerides at the 2x different levels, right? So first level is a tissue organ level, means I already explained a little bit, the different tissue organs, they metabolize glucose differently. The glycogen is only being stored in the liver and muscle, and only liver and kidney are able to make glucose through the gluconeogenic pathways, right? Um, and different organ and tissues, they metabolize glucose differently. So we need to understand at this level. And another level is cellular levels. Cellular levels mean that each organelles or compartment, they have a specific roles in the regulation of metabolism, right? For example, the mitochondria. Have you guys know mitochondria, right? How important mitochondria? Who can tell me what is the main function of a mitochondria? Why our cell has a mitochondria? For what purpose? Yes. Yes, produce ATPs, right? It's the energy plant. So mitochondria is very important. It plays produce ATPs, and also very important place to oxidize fatty acids, right? So remember I mentioned the red blood cells were not able to utilize fatty acids because red blood cells do not have mitochondria, right? So not able to oxidize fatty acids. So different organelles have different, functions. So we also need to understand metabolism at the subcellular levels. Okay, so and the other important concept before we go to the details of metabolic pathways, we need to know is that the anaerobic pathways and anabolic pathways and catabolic pathways and metabolic pathways. What I mentioned. Anabolic pathways mean that the pathways that can synthesize the large components or molecules from smaller precursors, right? For example, glycogen synthesis, right? Glycogen is very complex, similar to the starch. The glycogen usually in the form of triglycerides stored in the animals, right? Starch is in the plant. So glycogen is very complex molecules that synthesized from the simple glucose molecules, right? So this glycogen synthesis pathway is considered anabolic pathways. And catabolic pathways is the pathway that break down large molecules into small molecules, like glycogen breakdown pathways, right? You break down the glycogen to release the free fatty acids, and this pathway is considered the catabolic pathways, right? And metabolic pathways basically is the pathways that link both anabolic pathways or catabolic pathways. So example, the citric acid cycles or TCA cycles as it considers this specific and metabolic pathways, right? They connect, these two anabolic or catabolic pathways. And the other concept is the aerobic pathway or anaerobic pathway, or aerobic metabolism or anaerobic metabolism. So aerobic metabolism basically is the aerobic means the required air, means that this pathway requires oxygens, right, to oxidize the nutrients. And as So where do you what do you think of where does the aerobic metabolism occur in the cells? We just mentioned the important organelles, mitochondria, right? Mitochondria is very important organelle to oxidize fatty acids, but also the glucose to be complete oxidized, right? And this process requires oxygen, right? So anything that can be oxidized in the mitochondria, this pathway is considered aerobic metabolism, requires oxygen. So aerobic metabolism requires two things. One, oxygen, and the other thing is mitochondria, right? Mitochondria is placed to oxidize, and oxygen is required for the oxidative reactions. And so what are the end products of aerobic metabolism? Requires oxygen and complete oxidized in the mitochondria. What do you think the product would be? See that again? Water and CO2, yes, thank you. Water and CO2, right? Complete oxidized or glucose or fatty acids will generate CO2 and water, right? This complete oxidized aerobic metabolism. And anaerobic pathways or metabolism means that this metabolic pathway does not require air or oxygen, right? And also you don't need mitochondria, right, for oxidation. So where does anaerobic metabolism occur in the cell? What do you think? Cytosol, right? We don't need mitochondria. We'll be able to metabolize glucose through the anaerobic metabolism. And what are the end products of anaerobic metabolism? Lactate, wow, you're excellent. Lactate, right? Lactate are the end product of anaerobic metabolism of a glucose, right? What do you guys think fatty acids can be metabolized through anaerobic pathways? No, right? The glucose is able to be metabolized through the anaerobic pathways and not fatty acids. Fatty acids need to be oxidized through the aerobic pathways in the mitochondria, right? Okay, so also I have two or maybe three slides of this table that I made. I think I just listed a couple of things that you guys probably wanted to know, to remember, right? So in terms of metabolic pathways of glucose, we're going to learn many. 2, 3, 4, 5. 1, 2, 3, 4, 5. Yes, 5 metabolic pathways of glucose. And you need to know whether these are the catabolic or metabolic pathways, and in which physiological conditions and these pathways are activated or more active, right? And in which tissues these pathways occur, and what is the function of this metabolic pathway? Why our body to do glucose metabolism through this glycolysis or gluconeogenesis or whatever the other pathways, right? What is the purpose or function of each metabolic pathways of glucose, right? And then each pathway has lots of biochemical reactions, and you don't have to remember, or maybe it's also difficult to remember all the reactions, right, in each pathway. But you wanted to know the rate-limiting reactions in each pathways. And a key enzyme that catalyzes this rate-limiting reactions, right? And how this pathway is regulated by hormones. So rate-limiting reaction are the reaction that actually control the whole pathway activities, right? And each rate-limiting reaction catalyzed by specific enzyme. So this key enzyme, if we can control the activity of the key enzyme, then we can control the key rate-limiting reaction, then can eventually can control the activity of or rate of the whole pathways, right? Does that make sense? So, and all this control or regulation is accomplished by the hormones. The most important hormones that our body secreted or regulates the glucose metabolism are the insulins, glucagons, right, and epinephrines. All these hormones are able to regulate the metabolism by controlling key enzyme activities and rate-limiting reaction activities, right? Okay, so now I wanted to tell you how our body be able to maintain blood glucose levels during fat condition or during fasting. And we know that blood glucose level can be fluctuate, right, during these two different physiological conditions. So our body be able to maintain blood glucose at very narrow region. For example, post-absorptive state, we probably have a little bit higher blood glucose levels, and during starvation, we have a little bit lower. And if we eat the high carbohydrate meals, and we probably even a little bit higher, right, the blood glucose level. But our body will be able to maintain this narrow range of blood glucose levels. And before we talk about the regulation of blood glucose levels, I wanted to explain a little bit more detailed of each metabolic pathways of the glucose. The glycolysis is one of the pathways. Glycolysis means it's a metabolic pathway that can convert glucose into pyruvate, right? So glycolysis pathway that glucose can be metabolized to generate pyruvate in the aerobic conditions. And glycogenesis is also called glycogen synthesis, right? Glycogenesis is same pathways as glycogen synthesis. So glycogen synthesis or glycogenesis is a pathway that glycogen is synthesized from the glucose, right? And gluconeogenesis. Gluconeogenesis is a pathway that can produce glucose from non-glucose precursors. And this pathway should be activated during which physiological conditions? You can imagine. In what a condition that our body needs to make more glucose in order to maintain blood glucose. The fasting, right? Fasting, when our blood glucose level goes low, then gluconeogenic pathway can be activated to make more glucose to be able to raise the blood glucose level, right? And the glycogenolysis is also called glycogen breakdown. So glycogen breakdown pathway is basically break down glycogen by enzymes and to release glucose. And this pathways is also should be activated during fasting, right? So two pathways, gluconeogenesis and glycogen breakdown pathways, should be activated during fasting. And glycolysis in glycogen synthesis should be more active during fat condition, right? Does that make sense? So you try just connect this pathway to the different physiological condition, right? Okay, so this just give you a overview of how dietary glucose is metabolized after meal. In other words, how our body metabolize glucose in order to maintain blood glucose level, to not allow the blood glucose level to go too high after meal. Right? We absorb a lot of glucose from the food. So, start with the digestion of dietary carbohydrates. So once we digest carbohydrate and we absorb the glucose into the enterocytes, right? In this, it occurs in the small intestine, and some part of the glucose can be directly used by small intestine, and most of the glucose being absorbed into portal vein and to the liver, and the 20% of absorbed glucose metabolized in the liver, and the rest of the glucose actually go to the circulation and to the other tissue for the for metabolism, right? So in the liver, this 20% of absorbed glucose can be utilized for the energy, go to the glycolysis, right? And a lot of glucose can be used for synthesis of glycogen and store as energy in the liver, right? And so in the fat condition or after meal, the glycolysis and glycogen synthesis pathways are very active in the liver. And other two pathways like glycogen breakdown or gluconeogenic pathways are not active in the fat condition because we have enough glucose from the diet, right? From the food. We don't need to break down, our stored energy. So also we don't need to make glucose. And another pathway is that if we really have a lot of glucose being absorbed in the body and we can't use it, or enough stored, and then the glucose can be converted to fat and stored in the body, right? So this all happened in the liver after meal, and also a lot of like 80% of glucose, non-uptaken or non-metabolized glucose, which is 80% of absorbed glucose, goes into the circulation and to the other tissues. And because, so here I wanted to mention that very important hormones, that insulin being released after meal when we have a lot of glucose absorbed. So blood glucose level goes very high, and high blood glucose level can stimulate insulin release from pancreas, right? So once insulin is released, and what insulin does is to stimulate the glucose in the circulation being uptake into the tissues. And skeletal muscle and adipose tissue are the two major tissues that glucose being taken up by insulin stimulation, right? So insulin can stimulate glucose to go into the two major tissues, skeletal muscle and adipose tissue. The reason why insulin only stimulates glucose into these two tissues and not the other tissues? Can you guys give me thinking about it? Why? Everybody knows? Group 4? What was this thing? Sorry. I saw you said group 4. No. So it's something related to glucose transporters, right? So skeletal muscle and adipose tissue are the two major tissues that express or produce group 4. We're going to talk about glucose transporter later, right? So group 4 is the glucose transporters that's regulated by insulin, right? So so this is the reason that why insulin only push the glucose go to these two tissues, because of these two tissues express group 4. Group 4 is insulin regulate glucose transporters. Right? And the other tissues, the 22% of glucose being uptake by brain, and the other tissues, right? So, and I wanted to mention that the 80% of insulin-stimulated glucose uptake goes to muscle, and 20% to the adipose tissue, right? So, means after meal, a lot of because insulin levels increase, so a lot of glucose being uptake by muscle and adipose tissue, right? So this is an important process to allow the glucose level being decreased, right? Now, how about during fasting? And we don't have a glucose, right, from the food, but we need to maintain glucose level in the circulation, because the brain is very important, and also red blood cells only rely on glucose for energy, right? So for this reason, that our blood glucose has to be maintained at the normal range. Otherwise, we have trouble with the brain function and also red blood cells. So during the starvation, and we have to use the stored energy, right? So stored glucose to maintain the blood glucose level. So first of all, glycogen stored in the liver being break down to glucose and released into the circulation to raise the blood glucose level, right? So this is an important source, and also the gluconeogenesis. If starvation goes longer, then just breaking glycogen not enough to maintain blood glucose level, then gluconeogenesis will also activated. So liver and kidney are the important organs that make that do gluconeogenesis, to make glucose from sugar precursors, right, and also contribute to the circulating blood glucose levels. And why I only listed the liver in here, not skeletal muscle? We know skeletal muscle also store glycogen, right? Why not muscle? Does muscle contribute to circulating blood glucose level? Why? Yes, you're right. But muscle does not release glucose. The reason, why the muscle is not able to release glucose? Yes. What's that? Not quite. They actually lack the very important enzyme that we're going to talk about later. The glucose 6-phosphatase is the enzyme that break down the glycogen and release the glucose. So because muscle does not have this enzyme, so not able to release glucose, not able to convert a glucose 6-phosphate to glucose and release it. Right. So just try to remember only liver glycogen that contribute to circulating blood glucose levels during fasting, not skeletal muscle, right? Okay, now let's talk about the glucose uptake by tissues. We already mentioned a little bit, right? Is the key regulated step controlling glucose homeostasis, right? During the fat condition we eat, and we push a lot of glucose being taken up by tissues, so then can reduce the blood glucose level. And so glucose transporter activity is very key to maintain blood glucose level, right? So we have 5 different transporters that have been well characterized so far, but of course there are more than 5 transporters has been discovered, probably more. So for this class, we just wanted to remember the 5 different transporters that are responsible for transport glucose, and different tissues that express at different transporters, and they have different functions. I just wanted you to remember just the 2 categories. One is called insulin dependent, and the other category of glucose transport insulin independent. Insulin dependent and insulin independent glucose transporter. So it should be very easy to remember, right? Only group 4 is the insulin dependent, and the rest of the transporters are insulin independent, right? Insulin dependent means group 4 only when insulin level is high, so then group 4 activity can be high, can be activated. And the other glucose transporters are not regulated by insulin, but by glucose levels, right? So here are the table that listed the 5 the different transporters, and the major site means that the expression levels, right? And I want you guys just try to remember this, right? Group 2 versus group 4, and group 5 and SGLT1 you already know, right? The group 5 is basically in the small intestine that transport fructose, right? And so the fructose, and SGLT1 is small intestine, and is also important for glucose transporter into the enterocytes. And group 2 and group 4, they have a different distributions. The group 2 basically expressed highly in liver and pancreas, and also small intestine, kidney, and responsible for glucose transporter. And group 2 is a glucose regulated transporter, meaning that group 2 activity is only regulated by blood glucose levels. When blood glucose level is high and group 2 activity is increased, so then can transport blood glucose into the liver or pancreas beta cell, right? And group 4, and again, 2 major tissues, skeletal muscle and adipose tissue, right? The major sources side of group 4. So group 4 again is insulin stimulate glucose transporter. So only insulin stimulate glucose, group 4 activity increase in the fat condition. So how glucose transporters regulate glucose metabolism? In the fat conditions, that the 2 main glucose transporters are involved in the glucose metabolism, group 2 and group 4. Group 4 again is insulin dependent, right? The insulin stimulate group 4 go to the skeletal muscle and adipose tissue, and group 2 is insulin independent, right? And liver and pancreas are the 2 major sites that produce a group, that express or produce a group 2. And group 2 is the, again, the glucose regulate dependent transporters. Right? Any questions so far about the glucose transporters? Okay. Yeah.
So you said that really only
That's a good question. So group 5 is the transporters responsible for fructose, right? Uptake.
So if you had ate a lot of fructose,
Mm-hmm.
wouldn't that be considered fasting? That's taken out of
Yeah, I mean, but just because of a glucose is more abundant, because starch or when you digest starch, you just release a lot of glucose, right? So you have a lot of glucose actually being taken up. So here we talk about the glucose, and fructose is different, and not only in the amount, but our metabolism is also different. We're going to talk about it. And glucose be able to stimulate insulin release, but fructose is not able to stimulate insulin release. They're quite different. Um, so just because of abundance, I think glucose is more important in terms of the effect on the blood glucose levels, right? So here we just wanted to learn how GLU2 regulate glucose metabolism in the fat state. Again, GLU2 is expressed in two major tissues, liver and pancreas, right? When we eat the food, we absorb a lot of glucose and raise the blood glucose levels. And this increased blood glucose level can stimulate uptake of glucose by GLU2, right? GLU2 is more active when blood glucose levels high. So GLU2 and take up a lot of glucose into the liver and pancreas. And in the liver, we already mentioned in the couple slides earlier, right? In the liver, glucose being mostly synthesized glycogen and stored in the liver, and also can be converted to the fatty acids or triglyceride and as a fat, right, in the liver. And of course, some of glucose can be directly oxidized for the energy in the liver. And in the pancreas, the glucose uptake by pancreas or beta cell particularly is a very important stimuli or sensor, can stimulate insulin release, right? So the main function of glucose being taken up by the pancreas or beta cell is to stimulate insulin release, right? So insulin is very important hormones that regulate the glucose metabolism in the body. So now let's talk about the glucose stimulate insulin release. This is also considered the first tier response to elevate blood glucose level, right? So when we eat a food and we raise the blood glucose level, so immediately our first response to this high levels of glucose is to stimulate insulin release, right? So this is the first tier response is the insulin release from the beta cells. So this is the diagram to showing the process how glucose is able to stimulate insulin release. So glucose again transported by GLU2 in the beta cell into the beta cells and goes glycolytic pathway to metabolize and to generate generate ATPs, right? So once you have a glucose being uptake into beta cell and you produce ATP, you have to increase ATP to ADP ratio. So this increase can cause the open up of the potassium channel to allow a lot of potassium go out of cells and causing depolarization of the cell membrane, right? And depolarization of the cell membrane will activate the calcium channel to open up and allow a lot of calcium to be influxed into the beta cell and causing the increased calcium concentration in the cells. And this increased calcium concentration can trigger the insulin granule to move to release insulin, right? So this is a whole process, how glucose uptake by beta cell and to trigger insulin release. So again, try to remember this is a first year reaction to activate glucose, blood glucose after meal. Right? Is glucose, high glucose level to stimulate insulin release is the first year response, right? So here I have a question. We know that which tissue organ produces insulin is beta cell, right, in the pancreas. And second question, why insulin is produced? Why we need to produce insulin after meal? What is the purpose to have an insulin release, have insulin high? Yes. Yes, yes. Thank you, excellent. So we raise the insulin level and to allow the more glucose being taken by tissues, particularly adipose tissue and skeletal muscle, right? Then to reduce the blood glucose level. So the purpose of insulin release is to trigger the second tier response. That means to push the blood glucose level go into the tissue, skeletal muscle particularly, and adipose tissue, right, for metabolism, and then release the blood glucose levels. I guess, okay. Yeah, let's just finish this slide. So the first theory of response is that the dietary glucose causing the increased blood glucose levels and stimulate glucose insulin secretion from beta cell. And then the insulin is a very important hormone that triggers a secondary response. That means to stimulate the glucose uptake into the cell, right, and to reduce the blood glucose level. So secondary response is the it involves two things, right? One is insulin, and the second thing is the glucose transporters, right? Particularly by GLU4. So glucose transporter, which glucose transporters that involving the secondary response? That triggers by insulin. GLU4, yes, thank you, GLU4. So after meal, elevated glucose levels triggers first tier response, which is the insulin release, and then insulin starts the second tier response, right? So the GLU4 is involved in the secondary response. And what tissues? Muscle and adipose tissue, right? So take up glucose by GLU4. Right? Okay, so I think we just stop here with the lecture, and we have informal writing for today.